Hello, and welcome to Schmidt Ocean Institute's Back Ashore series, where we gather scientists who have sailed on board our vessels and learn what they are up to since they've disembarked from their oceanographic expedition. Today, we are not focusing on a scientific topic as we have for some of our past episodes, but we're focusing on a geographic location, the Gulf of California, Mexico. Excuse me. Schmidt Ocean Institute's research vessel Falkor conducted research in Mexican waters, specifically the Gulf of California, 3 times in 2018, in 2019, and in 2021. In fact, working in Mexico was the very last expedition of research vessel Falkor before it was donated to Italy. Today, we are joined by Dr. Samantha Joy, Regents Professor at the University of Georgia, who was Chief Scientist on board in 2019. Dr. Ronald Spelz Madero, Professor and Investigator at the Autonomous University of Baja California, Mexico, was Co-Chief Scientist in 2018 and in 2021. And finally, Dr. Manet Peña Salinas, also from the Autonomous University of Baja California, sailed in 2021. Thank you all for joining me, and I'm really excited for this conversation. We're going to go ahead and dive in to our discussion, but I want to remind everyone in the audience to put their questions in the chat, and we'll make sure our esteemed scientists have time to answer them. Ronald, we're going to start with you. You were on board during Falkor's first expedition to Mexican waters in 2018. Could you tell us a little bit about what the goals of that project were?
Yeah, thank you. Absolutely. Before I get into answering that question, I would like to thank you so much for the invitation to be part of this panel and to share this space with Manette and with Dr. Samantha Joy. So yeah, it's been exciting. And to answer the question, it is important first to set a little bit of context of the 2018 expedition. Before 2018, MBARI scientists conducted groundbreaking work in the Gulf of California in 2012 and 2015. So I was fortunate enough to be part of those expeditions as well. And at that time, there weren't hydrothermal band fields known between the gap located between Guaymas Basin and the East Pacific Rise in the south. So it was a gap that was largely unexplored in where no hydrothermal bands were known at that time. It's 750 kilometers. So we see the Guaymas Basin over there, right in the middle of the Gulf of California. And to the south, the red segment in the very far end bottom, of the image is the East Pacific Rise. So there were no hydrothermal band systems known prior to 2012 and 2015. So that changed during those expeditions, and they discovered 2 new band fields in the Alarcon Rise, which we can see there right above the East Pacific Rise, and then a little bit farther north in the Pescadero Basin. So the 2018 expedition above the Falkor was designed to build on that foundation. Our main goal was to conduct high-resolution seafloor mapping and explore the hydrothermal and tectonic systems of the Pescadero Basin in greater detail. So we named the hydrothermal system in the Pescadero Basin Auca, based on the human tribes that inhabited the peninsula during, ancient times. So using the AUV and using the ship's multi-beam sonar, we generated the first 1 meter resolution bathymetric maps of the North and South Pescadero basins. And additionally, during this 2018 expedition on the Falkor, we conducted the first centimeter scale mapping of the Auca band field using optical and acoustic techniques mounted on the ROV Sebastian. which allowed us to visualize fine-scale structures and chimneys, surrounded habitats, and with unprecedented detail. So this effort in 2018 led to the discovery of a new hydrothermal vent field, which we named Jakma. So this was located like 2 kilometers southeast of Auca. And we also carried out fluid sampling, ROV dives, and microbial and biological surveys during that expedition. So making it one of the most comprehensive and interdisciplinary investigations in the region to date.
Great. Thank you, Ronald. Lots of discoveries and firsts I hear in that expedition, and I think we'll get to more of that discussion later. But let's continue with our overview of these expeditions. Mandy, I know you have since been back to the region that you were studying in 2019, and we'll talk a little bit more about that in a bit. But could you start with telling us the goals and giving us an overview of your 2019 expedition?
Sure. Thanks, Allison, for the invitation to be here today. Ron showed you a moment ago a map of the Gulf of California system, and the Guaymas Basin, it sits in the middle. It was one of the first hydrothermal systems that was studied outside the East Pacific Rise when vents were discovered in the Pacific in the olden days when we only had Alvin to explore these systems. And Guaymas Basin has received a tremendous amount of work since then. But really, prior to our expedition in 2019, most of the work that had been done focused either on the sediments or on the upper part of hydrothermal plumes, these neutrally buoyant plumes that were moving laterally away from areas of fluid discharge. What we wanted to do on our expedition was take advantage of this rare combination of geological dynamics and biological response that happens in the Guaymas Basin, because you can think of the Guaymas Basin a bit. There's a sill on the outer, the bottom 200 meters of the Guaymas Basin is isolated, and in that section of the system, there are hundreds of hydrothermal vents, some enormous, some quite small. But this whole area is just incredibly impacted by this hydrothermal system. And we wanted to see whether we could actually track the connection and the biological imprinting of this hydrothermal venting on the biological system and the water column. People had done that in the sediments to some degree, but we really were aiming for super hot sediments. We wanted temperatures exceeding 100 degrees C by 10 centimeters, which are plentiful in the Guaymas Basin. And then we were targeting these hydrothermal plumes. And we wanted to get really close to the vents, as close as we could get without damaging the rosette. And we perfected the technique for basically catching these plumes with the rosette because of just exceptionally gifted pilots on the Falkor that could really drive the boat and shimmer and position these vents. And we got, I think, some of the best samples of, hot fluids, not super hot, 20 degrees above background temperatures, which are about 2 degrees in the bottom. But we got some just spectacular samples, which we'll talk about a little bit later. But the sediment part of this project was really successful, and we were able to show these connections between the benthos and the water column.
Excellent. Thank you for that description, Mandy, and thank you for the shout out to our wonderful and amazing crew and technicians on board the ship. They work really hard to make all this science happen. And finally, Manette, could you talk about the goals and objectives of the 2021 expedition, and what was your role on board during that expedition?
Sure, yes. First, I really want to thank for the invitation. It's really amazing to be here and sharing all about this science with these amazing scientists. So in 2021, we have to go back to the Pescadero Basin because, as Ronald mentioned before, the new hydrothermal vent field of Jakma has been discovered. So we really wanted to study it in a more integrated way, like combining the geology, the geophysics, and the biology approach. So the plan was to map the nearby basins of Carmen and Farajon. We also wanted to take rock samples, doing heat flow measurements, also taking biological samples, taking sediment cores in different places from the Pescaro Basin, from Auca to Jakma. And I was involved in different parts of the expedition that I was helping with the rock sampling. I was doing some heat flow measurements during the lake from geophysical data. But I spent most of my time working on the wet lab. I was processing the sediment cores. I was subsampling, taking samples for DNA extraction and for geochemical analysis. And I also helped with the outreach activities. I was connecting with schools from the ship, showing the things that we were doing on board, and which was obviously a great experience.
Thank you, Manette. Sounds like you had a multifaceted involvement in the expedition, just as much as the expedition was interdisciplinary. You had your hands in
Yes,
everything.
totally.
Great. Okay, so Ronald, we're going to go back to you for a second. You're the geology expert here, and while Mandy did do a great job of setting the scene of the Guaymas Basin, all of these expeditions took place in the Gulf of California, and I was hoping you could tell us a little bit more about this unique location and why it's the focus of so much research.
Yeah, absolutely. I mean, the Gulf of California is one of the most geologically dynamic places on Earth. It's a very young ocean basin. I mean, it's the tectonic activity began, at least 12 million years ago, but it opened up like around 6 million years ago. So it is very young. It's a tectonic rift that it's literally tearing the continent apart. So over the last 12 million years, the Baja California Peninsula has been gradually moving northwest away from mainland Mexico, opening space for the Pacific Ocean to flood in. So this rifting process is driven by the boundary between the Pacific and North American plates, which here connects the East Pacific Rise, as I mentioned before, which is a major seafloor spreading center. And it's connected through the Gulf axis with the San Andreas Fault system, which we cannot see in these images, but is a little bit farther north from Mexicali. But you can see the transition, along the Gulf of California, all the faults that are linked by these red segments, which represent spreading centers, such as the Guaymas Basin, the Carmen Basin, Farallon Basin, and other basins that are distributed along the axis. So it is a rare opportunity to study continental breakup and ocean basin formation in real time in this area, in this tectonic setting. But it is important to mention that this tectonic activity does more than reshape the Earth's surface. As the crust stretches and thins, it allows heat from the Earth's interior to rise. So the crust gets thin, and the heat from beneath the Earth, it's closer to the surface. And this high heat flow gives the Gulf of California tremendous geothermal potential, and not just for energy development, but also as natural engine powering life. And these hydrothermal vents that we have been studying in Guaymas, as Mandy said, and in Pescadero Basin, and also in the Alarcon Rise, support rich and diverse communities of extremophile organisms, microbes and animals that thrive really in complete darkness under extreme pressure and you know, their energy do not come from the sun, but from the inner, the Earth's interior. This image is really important that you can see all the seismicity. I mean, it's a collection of epicenters from 1970 to 2017 from the source is the Incorporated Research Institution for Seismology. And you can see that the main activity occurs along the axis of the Gulf of California. You can see the magnitude of the earthquakes by the size of each dot, and the color represents the magnitude, and the size represents the magnitude of the event. So what is interesting here is that even though all the activity is occurring along the axis of the Gulf of California, you can still see some activity, some dots along the western margin of the peninsula. So what this tells us is that the Baja California Peninsula, it's behaving like at the present as a microplate. So it hasn't detached completely from Baja California. It hasn't been attached completely to the Pacific plate. So it behaves like a small microplate, which makes it really, really interesting. And as I was mentioning before, you know, all the heat that is produced in this area, it's kind of a reminder of a deeper truth that I always tell my students, that geology leads and biology follows. And I don't mean this, I'm not saying this that in a bad way, but you know, throughout Earth history, geological forces have driven environmental changes. Sometimes these changes are gradual, sometimes are catastrophic, and you know, life has to adapt, evolve, innovate, or perish in response. So that's what makes the Gulf of California really a really small lab in order to study all these environments and the geology and everything that goes around it.
Thank you for that very interesting geology lesson, Ronald. Mandy, I saw you nodding your head, and I know you kind of gave us a setting of the Guaymas Basin, but is there anything you want to add to set the scene for our conversation and for the research that you did in this area?
Yes, and I don't have a geology degree, but I should, because that's what I did to fill up all my electives. And the way that I think about the Gulf of California is that it's a window to the past, and it gives us a vision of the future. Hypoxia in the bottom waters, large scale methane emissions potentially happening. In the past, whether that was a emplacement of large igneous provinces, which are widely documented as pulse methane sources to the atmosphere, and in the future that could be methane hydrate destabilizations as the ocean warms. So this system for me is a place that we can both learn about the past and predict the future. And I think that's a really nice way to think about it. And it's not even just the future of Earth, it's ocean worlds, because these extremophiles that live in the sediments and hydrothermal rocks in the Gulf of California tell us a lot about organisms and how they could survive on other planetary and extrasolar bodies.
Thank you. It's definitely a very unique setting. Now that we have talked about the geology, let's dive in a little bit into what each of you discovered and found below the seafloor while you were out on Falkor. Ronald, you mentioned that there were some new vent fields discovered in 2018. Can you tell us more about those to kick us off?
Yeah, absolutely. I mean, during the 2018 expedition, the 2018 and the 2021 expeditions, actually, we discovered and mapped new hydrothermal vents in the southern Pescadero Basin. You know, the bigger one, besides Auca, which was discovered in 2015, is called Jakma, which means liquid metal. Liquid metal. in the human language. So the name reflects from the shimmering appearance of hydrothermal fluids observed in a cavern, which was lined with calcite minerals, which was one of the most striking features that we encountered. So this field, the Jakma field, lies just south of the Auca Bend site, and both are aligned along an active fault system on the western margin of the basin central graben. So structurally and thermally, they are quite distinct. Jakma features tall chimneys up to 25 meters tall and focus venting with extremely high heat flow, reaching at some points like 120 watts per meter square in localized zones. So, but what is especially interesting in the is the fluid chemistry at both sites, which appears to be nearly identical. So suggesting that they may be drawing from a common deepet seated hydrothermal reservoir. So these two fields share a common reservoir, and this opened up new questions about surface connectivity and how bent fields evolve over time. So there are intriguing difference, as we have mentioned, in microbial communities between the two fields, but I leave that to Manette or Mandy, who are better equipped to explore that topic. But also what was interesting during the 2021 expedition in particular is that we detected, while mapping 13 methane gas flares across the Pescadero, Farallon, and Carmen Basin. I think so. These plumes are maybe thermogenic or biogenic in origin. So these rise hundreds of meters above the seafloor and in the water column. So this could point possible venting sites that remain unexplored. So this would be like a clear invitation for future exploration. So here we see an oblique image of the Carmen Basin and the flare, there with red and yellow image. So they rise like several hundred of meters from the bottom into the water column. So as I mentioned, we detected 13 of these methane plumes, and this could be either thermogenic or biogenic. So it would worth to take a closer look during future explorations to the Gulf. So yeah, this is incredible, and these are linked obviously to the geology of the area, and you can see like right in the middle of the Carmen Basin, there's one methane plume right from the middle, rising right from the middle, and this area are there's been spreading activity. So new ocean crust has been formed in these regions. So I wouldn't be amazed if this area could host more hydrothermal band systems, which are about to be explored yet.
Thanks, Ronald. I love that in your talk about discovering new features, you've inserted a call for more exploration and research needed. That's true science right there.
Right.
Manette, Manette, you recently had a publication and presented at the Fall AGU meeting about the thermogata diversity at these vent fields. Could you tell us what the significance of this diversity is?
Sure, yes. Well, the idea of studying Thermotogota came from my interest of studying thermophiles in Pescadero Basin. So when we were analyzing the microbial diversity and the geochemical data that we got in 2018, we saw that these particular bacterial group was significantly present in Auca and Jakma. So we decided to get a further study on it. And we saw that these different families are linked to different temperature regimes in the sediments. So it is clearly in the basin in between Auca and Jakma that there are temperature gradients in the sediments caused by the hydrothermal activity that is happening between them and between the sediments. So clearly these families are reflecting that, where the different families are used to this kind of temperature gradients. So we are showing, we are looking at how microbial communities are structured and distributed along the sediments and along the Pescadero Basin between Auca and Jakma. So it's really a glance to see how these microbial diversity are distributed along these fields and are reflecting all these temperature gradients. And of course, having the fact that we found different families from Thermotogora also points to high diversity in the system. So we are like very excited about this, about all the microbial diversity that could be and that is right now in Pescadero Basin.
Very exciting indeed. Thank you for that description. Mandy, the expedition that you led in 2019 sort of became famous for seeing these upside down pools, we'll call them. And I remember watching your reaction to the ROV coming up on one of them. And your awe was just priceless and something that I'll never forget. And the video of those upside down pools has now been used as part of art exhibits. And I know you recently returned in the Alvin to see them again with your own eyes. Could you walk us through that moment of discovery and how you felt and reacted, and also explain the scientific significance of seeing these features?
Sure. So these upside down mirror pools are created essentially by the intersection of superheated hydrothermal fluids, the fluids in this particular, in these mirror flanges around Big Pagoda, is around 380 degrees centigrade, and the regular deep water is 2 degrees, as I mentioned before. So the difference, that sort of interface of those two temperatures, it has this impact of it effectively creates a mirror because of the refractive interface between those two fluids. So, and that's also what causes shimmering water at the edges of these flanges as the fluid is discharged underneath and above. These flanges, I had been on expeditions where we had seen much smaller features. I mean, this one was around 3 meters across and about 4 meters out. And one of the reasons I was so astonished, it was just like, how is that hot, buoyant water possibly holding that flange up? Because you get a rock about this big and it's heavy, you're just carrying around the deck. And this is a massive flange. And my brain is just trying to figure out like how much buoyancy has to be generated by this water to hold this flange up. And no wonder there's all these crashed flanges on the ground. They basically collapse under their own weight eventually. But they're really magical. If you look, when we get down far enough, it becomes invisible and you can see the inside. And those little stars flickering in the back, that's pyrite, which is fool's gold. And there's just tens of thousands of little flickers of pyrite that you can see as you look through. And if you're trying to get a young kid excited about science and nerding out about temperature and chemistry, this is how you do it, because they're amazing. I mean, I think the only people who've watched the videos of these mirror pools more than me are my 3 kids, because I have all these, reels of deep sea video that they have to watch because we don't actually get regular TV. But they just sit and they watch it and they go back and forth. And just like we were doing on the ship, I mean, they just do it again and again because it's just fascinating. But the other, interest scientifically is as these fluids mix with bottom water. So one of the hydrothermal systems are driven by chemoautotrophic microorganisms. So these organisms, they don't do photosynthesis, they do this other process where they basically consume the reduced chemicals that is being delivered by the vent fluid, and they use oxygen or nitrate or iron or manganese oxide as their electron acceptor. So they breathe oxygen just like we breathe oxygen. Some of them breathe nitrate. But that sort of mixing phenomena, it's like how fast can microbes grow in response to these plumes? Well, if you spend some time watching the plumes discharge, so if you just park the RV or the Alvin, and let me tell you, it was quite something to see that from the Alvin 2 meters away. I mean, it's just, yeah. There was a student in the sub, and like she came up and just like, you know, I could die tomorrow and I would be fine. My life is now complete because this is unbelievable. But the discharge is so incredibly tremendous that if you think about the time scale of microbes, and you know, a fast growing microbe can basically double every few hours. You know, these fluids are moving up 10 meters in a few seconds. Yet if you look at cell counts or transcript abundance, these communities are literally responding at the scale that the fluids are mixing, which as a microbiologist boggles the mind. So it has a lot to teach us about how life responds to perturbations and you know, stresses, because every once in a while, you know, the water gets really superheated and some of the organisms can't survive that, but others are like, Yahoo, all of my competitors have been fried by the high temperature and now I can thrive. So it's a constant battle between your biological capabilities and your physical and chemical tolerances. So in terms of understanding, you know, survival of the fittest, this is where you want to do it, because this is where microbes get challenged unlike any other place on Earth.
Thank you for that story, Mandy. And we have a question from the audience that any of you can answer. But Mandy, you talked a little bit about the microbes and some of the biology adapting to these conditions that they live with. And someone is wondering, how do you deal with taking samples in these unique conditions and then trying to bring those up to the surface to study them under obviously very different conditions? How do the samples fare, I guess you could say, and are there certain maneuvers with the ROV, such as surfacing at a much slower rate, that might help the survival of your samples? Mandy, I see you're nodding your head, so I'll let you kick this off, and then Ronald and Manette, if you want to chime in, please feel free.
Sure. So we've built a lot of sampling tools and devices that allow us to collect hot fluids of various temperatures. So we have one that uses temperature 100 cm. They have two other tools that use temperature above 100 degrees centigrade. And you know, that's boiling up here for us normal people. It's really important, especially, I study methane cycling microorganisms, and for microbes that cycle dissolved gases, the concentration of dissolved gases is actually a function of temperature and pressure. So the deeper you are, the more gas you can get in solution, but the higher you are, the less gas you can get into solution. So it's this, you know, there's this contest between, you know, what wins, temperature or pressure. And this is an interesting comparison between Guaymas and Pescadero, because Pescadero is about twice as deep as Guaymas. So you know, the gas fields that the microbes are exposed to at the same temperature are quite different because the pressure at Pescadero is a lot more. So we have to be very careful when we're collecting these organisms. Now, we're not really, in Guaymas, we're not dealing with true barophiles. Like, barophile is an organism that loves high pressure. And typically, it's not until about 3,000 meters that you start kicking into, you know, barophilic organisms, so they won't even grow at low pressure. You can't even get them to do anything. But we do all of our enrichments, all of our rate assays, everything is done at in situ temperature up to 90 degrees C, in situ pressure, and in situ gas concentration. We run the gases on this really slick cavity ring down laser spectroscopy system. It takes a minute per sample. So we can determine the field, the chemical environment of our samples within about 20 minutes of hitting the surface. And then we can recreate that in our lab van on the ship, where we can put everything at pressure and run our experiments. And we do, back here in Athens, I have these really enormous pressure vessels that we can do and that we use for enrichments. You can't do that at sea because they weigh 500 pounds each. Like, you can't really take them on a boat. But we have 4, 5 of them here that we use for, high pressure enrichments. And it's amazing because, before I had these high pressure systems, it could take many years to get like one organism to grow. We'd be like, oh, they turn over every 6 months, and then we grew them in the high pressure incubator, and it's like, oh, actually they double in about 5 days. And they just weren't very happy how we were incubating them. And that's the high pressure system's the only way to do these incubations and get in situ gas concentrations, because you can't squeeze blood into a turnip, and you can't squeeze gas into a fluid at 1 atmosphere.
Thanks, Mandy. I'm glad you've figured out how to keep all the microbes happy at the surface. Manet or Ronald, do you have anything you want to add?
Yeah, I would like to add, something about not microbes or not biology samples, but, fluid samples, which were really
Definitely.
interesting to collect. And as Mandy said, I mean, some of the bottles that we have, they have to withstand, really high pressure because, for example, in the Pescadero Basin, the depth of this basin, which is the deepest in the Gulf of California. And actually, I might say that the Jakma and Auca bend systems are the deepest explored and known hydrothermal bend system in the Eastern Pacific. So they are at around 3,600 meters deep. So you can imagine the pressure that it's at that depth. It's really incredible. So when you want to collect some fluid, you have to bring down bottles that are empty and you want to fill them up, in the bottom. So they have to be made by a specific material that withstand that kind of pressure and that kind of temperature, and this is titanium. So these are titanium bottles. So they are, perhaps the audience might be familiar with titanium. So it's the material with which they build they shield the spacecraft while re-entering into the atmosphere to withstand, high temperatures and stuff like that. So it's kind of the same material. And it is important because, it is like getting these samples or fluid samples. It is like when we go to the medic and they take our blood out for a blood sample, and they can diagnose different conditions based on the chemistry of our blood. So we do the same with the Earth. So we take these samples, the fluid samples, with these special bottles, and then do the geochemistry analysis, and then try to interpret based on the geochemistry what the conditions are in this particular site and how it compares to other sites, and get to know more details about the interior of the Earth. So it is really, really amazing as well, the technology.
Yes, thank you, Ronald. Thank you for bringing in the integrity of the water samples that must be obtained and maintained while we're bringing those to the surface. Manette, did you have anything to add?
Yes, in the part of biology, I would like to add that first, that shout out to the ROV Sebastian, that it's the perfect equipment to take biological samples. Also, the pilots that are helping us, the scientists, deciding which organisms are we taking up. So we have some collections on the deep of many organisms. And of course, there are some that we could not recover completely, but there are other organisms that could adapt and could resist to the temperature. I'm sorry, to the pressure change when we got it. When we got them on board. So, for example, with these worms, we try to analyze, or the scientists on board try to analyze also the DNA that are coming from the samples, and they are also doing some taxonomic descriptions from the worms. So they are very, very useful. And when we got these biological samples on board, the first thing that we do is deciding and choosing which organisms are going to be for taxonomical descriptions and which one others will be for DNA extractions and other molecular analysis. So we are able to recover some part of these organisms and to do further studies.
Excellent, thank you. And we have one more audience question. Manette, I think this one is directed at you when you were speaking about your paper, but why are the extremophiles so important to study, and how can students get involved in these kinds of studies? I know Mandy talked a little bit about just kind of the excitement and how we can get the next generation involved, but if you'd like to touch on that as well, please feel free.
Yeah, well, I really love extremophiles. Extremophiles are great examples of how our life in our planet works. So when we are studying extremophiles, we're also studying the limits of life in our world. So we are trying to study what is the maximum temperature that organisms, in this case, bacteria or microbial organisms, could live. But also about other factors like salinity, pressure. So we are really trying to study about how the extremophiles are living in our planet. So we are trying to understand a little more about the ecosystems that are living in our Earth, but also one of the approaches that we have about studying extremophiles is about looking for life somewhere else beyond our Earth. So if we are trying to find life in another ocean worlds, as Dr. Mandy said, we need to know where are the boundaries in life and where can we look up for. So studying extremophiles are giving us all the clues to trying to find some kind of life somewhere else. So that's one of the things that I really love about studying extremophiles. And also about for the kids, it's like we are studying all this amazing biodiversity in our planet, and there is a lot of organisms, animals, microbes that do very specific things that are very, very interesting. So if you're like you like a certain kind of organisms or very resistant organisms, extremophiles are the best way to look at them.
Thank you, Manette. I love that perspective about the ranges of life. So that was really great. Mandy, do you want to add anything?
Sure. I just extremophilic organisms, so organisms that live at high temperature, high pressure, low pH, high salinity, those organisms have specific biomolecules that allow them to thrive in those habitats. And some of them have special lipid membranes so that they basically can withstand, like at high salinity, they have to have specialized membranes so that the cells don't literally collapse because the osmotic pressure inside and outside is very different. A lot of thermophiles have not only special lipids in their membranes, but they have special proteins that help them organize their DNA and stabilize their DNA. There's special proteins and other biomolecules that these organisms have. So extremophiles have been for a very long time topics of great interest in the biomedical and biotech world. If you want to talk about biological warfare, go to a microbial mat. They're, the yellow and the white mat are just like right beside each other, but they don't overlap and intertwine because they basically have this invisible fence that separates them. So they have all kinds of really interesting antivirals as well. And, the CRISPR-Cas system, people may have heard about CRISPR. This is what they're using to do gene editing now. That is actually the innate immune system. So microbes have a lot of, biotech and natural products that can solve all kinds of problems that are relevant to humans. So it's not just ocean worlds, it's our world and our own well-being that benefits when we study the deep sea and other environments where these extremophiles flourish.
Amazing. Thank you so much. I think you made a great pitch, both of you, for why we should study thermophiles or extremophiles, thermophiles and barophiles. So that was an amazing pitch. I'm excited now. So we're going to move on a little bit to what you guys are working on now and how the samples and what you've learned from those expeditions are still informing your research today. So Ronald, I'd like to start with you. What are you working on now from the 2018 and 2021 expeditions?
Yeah, absolutely. I mean, that's a great question, and I have to say that there's so much fabric to cut from all the material that we gather from these expeditions that, I mean, we have many years to come to do something. So we have to go through various specific things and then work on side projects. So what we're doing right now in the university, it's we're working on the South Pescadero Basin, which is actually not the South Pescadero Basin, but the whole complex of the Pescadero Basin, because as a result of our mapping, which I forgot to say that during the 2021 expedition, we mapped over 20,000 square kilometers of ocean floor, which is equivalent to around 1/8 of the total surface of the Gulf of California. So it is a huge amount of material or terrain covered during this expedition. And here in the image that we are looking at right now, it's the integrated map that we collected. It is overimposed under lower resolution map, which is coming from GeoMap app. So we are overlapping our mapping, our high resolution mapping, and we can see the yellow dots with the red dot in the center are the methane sites that we detected during the mapping operations during the first leg of our 2021 expedition. But the yellow frame actually portrays the whole complex of the Pescadero area. So before this campaign, the Pescadero area was divided into only two basins, the northern basin and the southern basin, which perhaps the letter over there in the map are a little bit small, but we can see the northern Pescadero Basin, NPV, and then on the south, the SPB is the southern Pescadero Basin. And then there's the central Pescadero Basin over there. So all these basins collectively are called the Pescadero Complex. And this is a really exciting place because, we have data available from other expeditions that have done seismics across this area. So we have like the structure, it's a window to see the structure or what is below the surface of that area. So we can see how the faults are, going through the crust, how deep they're reaching. And what we have learned about this area is that there are being two pulses of seafloor spreading. So this particular area has been significantly prolonged compared to adjacent sections. The stretching across the Pescadero Basin was significantly prolonged compared to adjacent sections along the plate margin. So this suggests that the material portions of the continental margins have been squeezed into the region of separation. So we know this again because of we have the seismic images, and this is counterintuitive because this process has counteracted the effect of stretching. So instead of thinning the crust, it has thickened the crust with material from the lower continental crust. So that's a project that we have been working right now. We're working on the corrections of a new paper, and alongside this study, we are perhaps Manette can get deeper into that because there's a master's student and another PhD student working with data, derived from this expedition. So we have, which is really, really nice to say that, because a lot of students that had began their studies before the expeditions, they got their degrees with the new data that was collected during this expedition. So right now we're working with two new students. One is Isabella. She was actually on board the 2021 expedition, and she's working on the deeper on the heat flow, actually with Raquel Negrete, who is the expert on marine heat flow. So we aim with the study of Isabella to deepen our understanding on heat flow in the Pescadero Basin by analyzing and modeling the new data collected from the 2021 expedition. So she aims to dig deeper into the mechanisms of heat flow dissipation and interconnectivity between both fields. And there's Carlos, who is doing his master's in ecological functional ecology. But I will let Manette to talk a little bit deeper about that part, because she's the biologist here. But yeah, that's the main thing that we're doing right now.
Thank you, Ronald. Sounds like you have been very busy, but I appreciate the reminder that when you walk off the vessel, you're armed with years and years of data and analysis to go through. So sometimes we forget that we wave goodbye and we go on to the next expedition. But my favorite part is hearing that you're still working on the data years and years afterwards and what you're investigating. So thank you for that. And I'm glad to hear that new students are working on the data as well.
Yes.
Manette, you were in fact a student when you were on board Falkor, and you've since completed your PhD. So congratulations on that. How did the expedition inform your dissertation and your career, and what are you doing today?
Well, thank you very much. Also, thank you to the Schmidt Ocean Institute, because you have been part of the journey finishing my PhD. So I'm very excited. Actually, I was just at the middle of my PhD when I was on board on 2021. Well, I have studied hydrothermal systems through books and papers, but being on the ship, taking part in decisions, doing the sampling, the collections, and analyzing all the sediment course, it's really an experience beyond anything I have ever imagined, and I'm very, very happy about it. I was really excited when I was looking at the hydrothermal vents through all these cameras in live in the videos. So it was truly, truly amazing experience. For my PhD, mostly I work with data from 2018, but the 2021 cruise helped me really to gather all the information and to understand the context of where these samples are on all these data were coming from. So it's really different, like looking at only the nodes, but being on the ship, looking all the context, all how hydrothermal vents are and how the microbial mats are around them, all the organisms, it really gives you the big picture. So this really helped me to have a better understanding of how this hydrothermal system works. I really understood better the methodology that I was working with. And right now, also this experience helped me a lot with all the international collaborations that are having coming. So I'm very, very, very happy about it. And also, it has been really important for me for expanding this line of investigation in the university. So as Ronald mentioned, that we have new students, and that's very, very exciting, because all the data that we have, we gather from 2021, and also that we have been studying from 2018. I mean, the paper, it's only a glance of it. And now we have been working with Carlos, with Isabella, and more students that are working in different areas. Specifically, as Ronald mentioned, Carlos is working in his master's thesis, and he is proposing using functional diversity to study all the animals that are living beneath the sediments between Auca and Jakma. And we are trying to figure out if there is a relation with the heat flow and other the environmental variables that are around. So we are trying to use all the information that we have from 2021 to keep studying them. And also I'm working with the microbial diversity that we are finding from the 2021 sediment course. So I think that it's a lot of things that are still coming for Pescadero Basin, that are a lot of things that we're still going to discover.
Awesome. Please keep us updated on the work of those students, and congratulations again on finishing your PhD. Mandy, I don't want to jump the gun and reveal any of your outcomes, but when I last saw you in person, you mentioned some upcoming publications about your expedition. Is there anything that you're able to share with us about your recent outcomes?
Sure. So we have a paper that's in press in Nature Communications. It should be out in the next couple of weeks. And it's about, I mentioned earlier that most of the microbes associated with hydrothermal vents across the globe, generally speaking, are chemoautotrophs. That means they use chemical energy. They oxidize things like sulfide or hydrogen, and they fix CO2 just like green plants and phytoplankton do in the ocean. Guaymas is a strange place, because, and Pescadero as well, the entire Gulf of California, actually. It's not just these inorganic chemicals that are being discharged from in the hydrothermal fluids. It's also complex hydrocarbons like oil, methane, which is, the simplest organic carbon molecule, but also juicy low molecular weight organic compounds like acetate and methanol. And a student who defended and graduated back in 2021, Andy Montgomery, is the first author on the paper that we have coming out that actually reports the first documentation of heterotrophy in hydrothermal plumes. And the shocker to me, like, I thought it was going to be important, but it's so much higher than autotrophy in these fluids. It's astonishing. And it's not, we did multiple casts from, Guaymas Basin, and we did similar experiments in Pescadero on our 2024 cruise, but the paper that's coming out is on the 2019 cruise. And it's really just amazing because, you know, getting those samples, this was not the first expedition where we tried to catch fluid. And we didn't get big. And now then, you know, we can get hydro warming regimes. Like we can go from 380 with the vehicles, and then we can do these profiles with the CTD rosette. And when we do that, we see that, you know, activity almost responds instantaneously. It's really amazing how fast the organisms are able to respond to this injection. But it's not just autotrophy. There's a lot of heterotrophy as well. So that paper will be coming out in the next couple of weeks. And we have another one that is in revision that talks about methane oxidation specifically, and the vents in Guaymas, above Big Pagoda, the place we saw the video from. The methane oxidation rates in those 100-degree fluids are the highest that have ever been measured anywhere on Earth, including the Deepwater Horizon oil well blowout in the Gulf of Mexico, which used to be the highest rate ever measured anywhere, 6 micromoles per liter per day, which is a really high rate for methane oxidation. Usually it's 6 orders of magnitude less than that. But in Guaymas, it's 32 micromoles per liter per day. And it turns the methane pool over in a matter of a couple of weeks. So that, and this is the case for all depths less than 200 meters. So it's, I mean, when we talk about specialized communities that have adapted to really high concentrations of methane and other reduced metabolites that are injected with these fluids, I mean, Guaymas is really the place to study this. And Pescaderos is the response there is different because it isn't this isolated basin, and the physics, it mixes. It seems to mix more efficiently in Pescadero, so you don't have this like chemostat effect where the community is just getting juiced over and over and over again. So it's, you know, that the methane paper is also authored by a PhD student. She graduated 2 years ago in 2023. And, you know, both of them ended up, oddly enough, at Montana State. And I don't know whether that's the skiing or Yellowstone. I think a little bit of both. And the amazing people that, you know, they're working with there. But they both came to my group, you know, interested in the deep sea, excited to go on expeditions, but not necessarily committed to the nitty-gritty everyday challenge of working with extremophiles, which, as Manette can say, it's hard. And they both left, you know, with green thumbs, and, you know, they're both going to have pretty high profile papers on some of the organisms that they've isolated too. So I'm just really happy for these students. It's, you know, sailing on the Falkor is a life-altering experience for these students. And, you know, I'm really grateful to the opportunity that the Schmidt Ocean Institute has given us to work with, you know, their incredible teams at sea and through the outreach work that you all do. You know, I'm a huge fan of it.
Thanks so much for that, Mandy. I really appreciate it. I'm sure your students also had an amazing leader there at University of Georgia, with you mentoring them and guiding them. But I'm really glad to hear that they're staying in the ocean world. I think we're a little bit over time, but I want to give all our speakers kind of one last chance at parting shots. Anything I didn't ask, anything else that you're excited to be working on right now that's not necessarily Falkor related, or anything else that you would like to share before we wrap up. So Ronald, I'll start with you. Please feel free to share what you're doing today or what you're excited about.
Yeah, thank you. Thank you very much. And again, as Mandy said, I'm also a huge fan of the Schmidt Ocean Institute. When I have the opportunity, I give the website to my students and then to their colleagues and friends, family, so they can all engage in what you guys are doing, not only in Mexico, but around the world. So it's an amazing endeavor. And yeah, we do appreciate all the work and the hard work that you guys do in favor for the for research and the scientific community. And talking about that, I would like to take a moment to invite the audience and invite you guys to the upcoming Cordilleran section meeting of the Geological Society of America, which is, we are organizing this time April of next year, 21st to the 24th of April of next year. It will be hosted for the third time in Mexico. So there's only 3 times with this one that this meeting will be hosted in Mexico. The last 2 were in Puerto Vallarta in 2002. The other one was in 2012 in the city of Querétaro. And 13 years later, 2026 in Loreto, Baja California. So it is in the Gulf of California. So it is a great place to share all the scientific research of the Gulf of California as well, and all the Cordillera from Canada, United States, and Mexico. So we will be hosting that event in April next year at Loreto. So we will be happy if we will launch later the website, and it will be exciting if some of the audience and all the researchers can join us there in Loreto. There will be exciting field trips. The geology, it's amazing. The place is really exciting. And well, it's geology mixed with oceanography and tourism and everything. So it's a whole package. So I appreciate the opportunity to spread the word. And I would like to thank again for the invitation. It was really, really nice to share this space with you guys. Thank you so much.
Thank you, Ronald. That conference sounds amazing. I'm sure there will be lots of great scientific presentations, including some from Falkor, and I appreciate you sharing that. Manette, would you like to share anything before we end the session?
Well, I really want to thank everyone for being here and for having me. It has been really life-changing being on Falkor and doing all these amazing expedition. I'm very happy that after I got off Falkor, we still have a lot of information, and I'm very happy that we have also have worked and have done different outreach materials. So we have been sharing all the science that we did on Falkor in 2018 and 2021 here at the university and in different places where I have been like giving some talks and conferences. And it's very exciting how the audience, the public gets really amazed by these hydrothermal systems. And I'm really happy to share here in Mexico, in our country. So it's also like very amazing how we have these incredible ecosystems very, very near for us. And studying these ecosystems is really amazing for me. For the future, I'm still planning on working on them. And I have this astrobiological approach to study these hydrothermal systems. So I'm very, very interested on studying other ocean worlds and comparing with hydrothermal vent systems, in particular in Gulf of California. So I think there is a lot of science coming still. So thank you very much for that.
Great. Sounds like you'll be busy. And thank you for all of your outreach efforts. Outreach is a very important part of the research that is done, and I appreciate your work. And please let us know if there's anything we can ever do to help to support you. And Mandy, last but not least, we're very excited to read your upcoming publications, but is there anything else that you would like to share that you are working on before we end this back ashore?
Sure. We have so much work ongoing from our recent expedition last year, and we're still doing a lot of work with our 2019 samples, actually. But I wanted just to sort of end by giving a plea for the study of the deep sea. And I'm preaching to the choir, to the Schmidt folks, but when we've been talking a lot about the Gulf of California today and the systems that we've been fortunate enough to spend so much time studying over the past 20 to 25 years. But when you think about the deep sea as a whole, you know, less than 5% is mapped at high resolution. Human eyes on the bottom with an ROV or a human occupied vehicle, it's less than that. And we discover new types of hydrothermal systems on average about every 10 years. You know, it's only been going on for 40 years, but we went from, you know, East Pacific rise type systems to the ultramafic systems on, you know, the mid-Atlantic ridge. And then there are really bizarre sulfur-driven systems in back arc basins. Many were described and discovered first by the Falkor. But we have so much more to do in the deep sea and so much more to learn. And it's not just about going to sea for the sake of going to sea. I mean, these environments and the organisms that they support have a lot to teach us and to help humanity. Whether, you know, the cure for cancer could be in some microorganism that lives in a cold sea system off Peru. You know, we don't know, but we need to be looking. And the deep sea is a place, I as an oceanographer who studies the deep sea, when people talk to me about space and they're so excited about, you know, looking into the stars, I always turn around and say, well, don't you wonder about what's under the water out there and all these places that we haven't visited? You know, what I would like to see by the end of my career is as much money being invested in exploring the deep sea as we spend to explore space, because it's on our planet and it has so much to teach us. I want people to realize that we've only scratched the surface a teeny tiny bit. We have so much more that we need to do.
Thank you so much, Mandy, for those remarks. They were really great, and I will not try to top them because I know I can't, but I really do appreciate those closing comments and that sentiment that the deep sea is very important from the direction the plates are moving and what is happening geologically that Ron kind of opened this session with, all the way down to the extremophiles and the microbes and everything in between that we've talked about today. So I want to thank our scientists again, Dr. Ronald, Dr. Manette, and Dr. Mandy, for speaking with us. And thank you to everyone online for listening to this session. And we will be back soon with another session with scientists. So thank you again and have a good